Hi and Assalamualaikum. My name is Amir Hasman and my roommate is Wan Anis Izzati, Shazwana Shamira, Chu Pesin and Shafirul Razi. Today, I want to present about biofuel production from wheat straw by using fructose and glucose. Wheat straw is a leftover from harvesting grain has potential to be a big source of bioethanol form. Every year, a massive amount of wheat straw is produced globally with a significant portion going unused. This unused straw could be converted into bioethanol instead of relying on crops that compete with food production for land. Unlike bioethanol from food crops, wheat straw is a second-generation biofuel and does not require dedicated farmland. It can be broken down into ethanol through a three-step process. Researchers are looking into ways to improve wheat itself to make it more suitable for bioethanol production. This could involve breeding with varieties with more sugar or that are easier to break down. In this study, highlight wheat straw as a promising biofuel source. Different wheat varieties showed surprising variation in free sugar content, which is ideal for easy conversion to ethanol. Researchers suggest breeding super straw wheat rich in this sugar, potentially creating a sustainable biofuel option that doesn't compete with food production. There's a few key methodology that we use in this experiment, which is the first one is the DNS assay. The DNS assay is used to measure the amount of readily available sugar in wheat straw. The sample is the first diluted and mixed in a special DNS reagent. This mixture is then boiled alongside a solution containing known sugar concentration. By comparing the light absorption of both the sample and the standard at a specific wavelength after boiling, we can determine the concentration of reducing sugar in the original straw using a calibration curve. This provides a measure of the total amount of sugar the straw readily has available for conversion into biofuel. The second one is determination of FSS and starch quantities. It is to measure the free sugar in the wheat straw liquids. We use a DNS on half the sample without any prep work. The other half was treated with enzyme to break down starch into sugar. The difference between the two measurements gave us the amount of sugar specially for starch. The third one is ion chromatography. Researchers use a powerful technique called HPA-EC PET to analyze the specific sugar in the wheat straw liquid. This involves separating the sugar with an electric current and comparing them to known sugar standards. They employ two methods, a gradient elution to identify a wider range of sugar and an isocratic elution to precisely measure the amount of glucose, fructose and sorbitol present. Last but not least is thermogravimetric analysis. It is to analyze the straw's decomposition that researchers perform this method. Tiny samples were gradually heated to 800 degrees Celsius while measuring weight loss. This allowed them to pinpoint the temperature at which different components within the straw decompose based on the rate of weight change. Researchers analyzed the sugar content of various wheat straw for bioethanol production. They found a water-soluble fraction rich in free sugars and starch ideal for conversion to fuel. Interestingly, domesticated wheat had more water-soluble fraction sugar than wild varieties, with fructose being the most abundant. While high sugar lines showed a distinct thermal signature, with the main components like cellulose and lignin did not differ significantly, suggesting another factor influencing sugar content. The amount of free sugars in wheat straw sample we studied varied a lot. These sugars are simple sugars that can be easily fermented into ethanol using existing yeast. The good news is that extraction this sugar from the straw is very cheap and easy. Even better, this sugar extraction process is so simple. It could potentially be done by farmers themselves on their farm by allowing them to produce bioethanol on site. While there are some difference between wheat varieties in terms of how easily the cell wall can be broken down for sugar extraction. This seems like a promising avenue for further research. In conclusion, both increasing the amount of free sugars in wheat straw and making the cell wall easier to break down would make the wheat straw an even better source of bioethanol.